Okay. Let's go ahead and play. Photosynthesis, light reactions, with a bonus question at the beginning. From the organism of the day. Do you remember the family? Nice. Yeah. So with all the plants, we're giving you the family because there's so many species that learning to group them into families can be a really useful way to organize the information. So the mint family is the Lamiaceae, and that includes the genus Salvia. Poaceae, you should know, is the grass family, and Fabaceae, you should know, is the legume or the bean family. And I think we did one orchid. So orchids similarly have flowers that are sort of elaborate and have a left and right symmetry, what we call zygomorphic symmetry. And so superficially, they might look a little bit kind of like a mint, but mints have a different structure and are much more common in the prairie than orchids are. So the Poaceae and the Fabaceae are two that you definitely want to know, and you'll encounter some mints. And you get to know the mints really well when you crush them up and sniff them, because they usually smell fragrant. All right, next one. Where does most plant biomass come from? Where do the plants get it from? Nice, good. Most of it comes from carbon dioxide. They do extract electrons from water, and they do transpire water, but they're mostly just moving the water from the soil to the atmosphere. It's just a conduit to move water, and they exploit that, the ability to move water as a way of trading water molecules for carbon dioxide. Soil is where nutrients come from, but most of the weight of a plant's body is carbon. And when you talk about biomass, usually that's measured by first taking the organism and drying it out so there's no water in there at all. And then you weigh what remains, which is the carbon. And then that carbon comes from carbon dioxide. All right. What are the inputs to the light reactions? The first part of photosynthesis, the only part we talked about last time, what goes into the light reactions? Okay, good. So I picked these first two because I wanted to remind myself to clarify something. It is in fact water, light, and then these two molecules, ADP and NADP plus. These two molecules carry energy, but these are in the form before, let's put it this way. The energy that's taken from sunlight is used to add phosphate groups to these two molecules that make adenosine diphosphate into adenosine triphosphate. When you add that phosphate, you're kind of storing energy, you're charging the batteries. So we have water, light, and then these are the molecules that are going to store that energy from the light. Oxygen is one of the products of light reactions. It's what's released when the water molecule is broken up. And then carbon dioxide is one of the inputs to the second part of photosynthesis, the Calvin cycle, and that's what we talk about today. So the first part is all about capturing that light energy and charging the batteries, creating that ATP and NADPH. And then those molecules are used to fix the carbon dioxide that we'll talk about next. Where do the light reactions take place? And I want you to think as specific as you can here. Okay, good. So chloroplast is not incorrect, but as you'll see today, we're going to be focusing on the thylakoid membrane, which is a structure in the chloroplasts. So chloroplast is right, but it's not very precise. On the thylakoid membrane is what we talked about last time, and we'll remind you of that again today. The thylakoid membranes, of course, being inside the chloroplasts. All right, last one. Nice. We ended with the Z-scheme. That was that diagram that we went through step by step. What is the Z-scheme? What does it show? This is one of those questions where we want you to pay attention not just to the shapes and symbols that you're seeing in the graph, but what do the axes mean? Yeah, good. Electron transport during the light reactions is correct. The second one, the blue one, was kind of tricky because ATP is synthesized as part of the Z-scheme. As the electrons are excited and move down the transport chain, ATP is synthesized, but it is not through the oxidation of glucose. Oxidation of glucose is what happens during cellular respiration. So if you didn't get all these right or you missed some of them, don't worry about it. The point is to inspire you about kinds of things that we might want you to know about. Nice. And Alex. All right. Paul, I picked up the recording when you got booted out, so I'll upload that at the end. Do you want to add anything about the bull snake story?
No, where did I get kicked off?
Right as I was talking about the prairie bull snake, and I pulled up this picture of what looks like a few students and a bull snake.
Oh, so they have a lot of bluster, but they're really quite tame. So they'll hiss at you and things like this, but if you just reach down and pick it up, I've never been bit by a bull snake, and they're as docile as can be. So
never been constricted by one?
No, not really. I mean, they're not that big. I mean, yeah. they're long, but they're not a heavy snake. So it's an easy snake to get to know. We used to have one in the biology department that lived on the first floor for a long time. So nice. They're nice snakes.
Okay. Sorry, I'm just getting ready to share the photosynthesis slides again. Here we go. Okay. So in our general schematic diagram of photosynthesis, we have the little bean shape here, which represents a single chloroplast inside the cell of a plant. And then within the chloroplast, you have these two parts to the photosynthesis reaction. The first part is the light reactions, and these little green stacks represent the grana that are formed by the folds of the thylakoid membrane. And it's among those folds in the grana where the light reactions take place, where ATP and NADPH are synthesized using energy from sunlight. Ultimately, this energy that's stored up here as chemical energy is used to power the Calvin cycle. If we could zoom in on the thylakoid membrane, I showed you this diagram last time. The thylakoid membrane has a whole array of enzymes and channels and photosystems built into it. So we've got these purple, pink, and green ones. Those are where ATP is synthesized. The ATP that's generated down the electron transport chain. The green, dark green, and light green represent photosystem 1 and photosystem 2. Those are complexes that have a whole lot of pigments in them, and those pigments are what capture photons and funnel that energy to the reaction centers where the electrons are excited, and then they can move down the electron transport chain, which is along the thylakoid membrane. So this blue one, for example, is a cytochrome protein, or a cytochrome intermembrane protein that's part of that electron transport chain that creates ATP. So the ATP synthase is also part of that, that process. I think I have an earlier one here where you can see the key, so you know what these different things represent. Photosystem 2, photosystem 1, ATP synthase, some of the key parts of the process. So once you got ATP and NADPH ready to go, we can think about what happens in the Calvin cycle, or the carbon fixation reactions. Here's a diagram showing generally the carbon fixation reactions. It is a complicated system, and it's named for Calvin because it was much of his career was spent on working out the steps involved in this reaction. But there's 3 phases to the carbon fixation reactions for the Calvin cycle that we want you to know about. The first one is fixation, or carboxylation. What's happening here is you've got these molecules called RUBP, ribulose bisphosphate, or sorry, ribulose bisphosphate, make sure I say that right. But the abbreviation that we'll use is RUBP. It's a molecule that has 5 carbon atoms in it. And there's an enzyme, ribulose bisphosphate, that's correct. It's a 5 carbon molecule. There's an enzyme called Rubisco. Rubisco is an enzyme that takes carbon dioxide molecules from the atmosphere and reacts them with that RUBP molecule to create 2 new 3 carbon molecules. So 5 plus 1 is 6, divided by 2, you get 2 3 carbon molecules. Those 2 3 carbon molecules are called 3-PGA. I'll show you a picture of that in a little bit. But the point is you go from a 5 carbon molecule called RUBP plus carbon dioxide, and you end up with 2 3 carbon molecules that are called 3-PGA. It's got a 3 there because there are 3 carbons in it. Then the chemical energy that's stored up inside these 3-PGA molecules is used to synthesize ATP, or sorry, the energy molecules that were synthesized by the light reactions are used to sort of add chemical energy to this 3 carbon molecule. So you go from a 3-PGA and you're sort of charging it up to get these new 3 carbon molecules called G3P. So you're adding energy to the system. That's the point here, adding energy to the system. I actually have I'm a little more comfortable doing this on the board, so I'm going to stop sharing in just a moment. But then these 3 carbon molecules are used in 2 ways. One of them is, you can think of it as being sort of set aside to make sugar. And for every 2 G3P molecules you set aside, you get a fructose molecule or a glucose molecule. And then the others that are synthesized are used to make more RUBP. This is called the regeneration phase. And you need to make more RUBP so that the cycle can continue. So let me show you what I mean on the board. I'll stop sharing for a second. Make sure you guys see me okay. so you start out with RUBP. This is the ribulose bisphosphate. This is the 5-carbon molecule that's used in the carbon fixation reactions. Rubisco is the enzyme that mediates the reaction that combines RUBP with carbon dioxide. So you get a total of 6 carbon atoms. And those 6 carbon atoms are apportioned into two of these 3 carbon atoms, or sorry, three carbon molecules, which we call 3-PGA. Now we've been talking about C3 and C4 plants for a while. This is where that C3 comes from. You take carbon dioxide, you fix it with the enzyme Rubisco, and you result in these 3 carbon molecules called 3-PGA. That's what defines a simple, or a typical, I should say, a typical C3 plant's photosynthesis process. It describes the first step of the Calvin cycle, or describes the carbon fixation step, maybe is a better way to put it. This is a really important reaction. This is how photosynthesis is powered, and Rubisco is the most abundant protein, seems to be the most abundant protein or enzyme on Earth, because it's what powers our biosphere, or most of it anyway. Most of the energy that comes into ecosystems comes through photosynthesis via this pathway. Few exceptions are organisms that get their energy from, say, geothermal sources. All right. So then through the process of reduction, these 3-PGA molecules through a series of reduction reactions become these G3P molecules. Chemically, it's very similar. A few bonds are a little bit different. It's still a 3-carbon molecule, but it's got that added energy through reduction from the energy that's brought in through ATP and NADPH. For every 5 molecules, for every 6 molecules of G3P that's produced, 1 of them goes to make sugar molecules, which are 6 carbon molecules. The other 5, 3 times 5 is 15, go to make 3 new RUBP molecules. Three new RUBPs. That way, you have RUBP ready to react with carbon dioxide through Rubisco and fix more CO2 into the system. So you can balance this equation out, and you find that an easy way to think of it is for 3 carbon dioxides that come in, you make, 9 of these 3-PGAs. Those are converted to G3P, and then 2 of those go off to become sugar, and the rest go off to be reconstructed into RUBP. But the point is that when you make this G3P, 3-carbon molecule, some of it goes to make sugar, and some of it goes to regenerate RUBP, so the cycle can continue. If you didn't regenerate that RUBP, you just use up the RUBP, and then the cycle would be over. Take a look here. So the 3 phases. carboxylation, or fixing the carbon, reduction, adding energy so that we can produce sugar, and then regenerating the RUBP so the cycle can begin again. Those are the 3 phases we want you to know. So when you're studying or going over this stuff, going over your notes and quizzing yourself, these are the kinds of questions we want you to be able to do. You should be able to label the parts of this diagram. You should be able to ask yourself, what are the inputs to the light reactions, just like we did with Kahoot. You should be able to ask yourself, what are the inputs to the Calvin cycle or the carbon fixation reactions, and what are the outputs? So who wants to remind me the inputs to the light reactions? Someone out there. The yellow zigzag and the blue circle.
Light and H2O.
Good. Yep, light and water. And what's that red circle, the output from the light reactions?
O2.
Yeah, O2. So what happened to the electron and the hydrogens?
It oxidizes the water molecule. Is that
So-
right?
You're close. So when the water molecule is oxidized, an electron is freed, right? It's made available for other purposes. And that electron goes into the photosystem 2 so that you can generate ATP. It adds energy to the light reactions and moves down the electron transport chain. The hydrogens, little protons, basically little protons or hydrogen ions, those occur along the thylakoid membranes and set up a proton gradient. And that proton gradient is also used in the synthesis of ATP. So the protons and the electrons that are made from oxidizing water are used to make NADPH and ATP, which are the 2 energy molecules down here. So before I hit the next slide, someone tell me what the input is to the Calvin cycle.
CO2.
CO2, good. And the main output, oh, I just clicked. Ta-da, it's sugars, carbohydrates. When these are soluble, they're available for reactions for cellular respiration, but sometimes we need to store that sugar and take it out of the system. So you can stitch a whole bunch of sugars together and make starch. And starch granules are a way that you can keep the concentrations of sugars low in the chloroplast so that the Calvin cycle can continue to produce sugar, right? If the sugar built up to high concentration, the reaction wouldn't proceed. So sugar can be packaged into starch, which is not soluble, and then the Calvin cycle can proceed. Now notice that when ATP and NADPH are used to power the Calvin cycle, they're returned to the system in a dephosphorylated state. So you've got these phosphate ions, and you've got ADP, so diphosphate, and NADP plus. So we add hydrogens back on here in the light reactions, and we add a phosphate group in the light reactions, and that's what gets you back down here to these energy carrying molecules. Okay. So let's consider for a moment how changing the environment, and in this case, we'll talk about brightness, influences the process of photosynthesis. So what we've got down here in the x-axis is the photosynthetic photon flux density. And you might remember that we talked briefly about this when we talked about incoming radiation from the sun. So this is a measure of the amount of photons that hit a square meter of surface every second. So photon flux density, photons per unit area per unit time. And then on the y-axis, we have one of the ways that we measure the process of photosynthesis. It's the rate at which carbon dioxide is assimilated or is taken from the atmosphere and put into the Calvin cycle. That is the rate at which it's fixed. So it's an amount of carbon dioxide per second. This is the typical shape of a curve that relates CO2 assimilation to light intensity or photon flux density. And I want you to notice a few things about it. The first thing is that the y-intercept is down here in below the origin. So it's in this negative area. And we call this the dark respiration rate. Can someone explain what we might mean by dark respiration rate and why it intersects here at the negative side? Take a stab at it. What's the opposite of CO2 assimilation? If you're taking in CO2, what's the opposite of that?
Giving off.
Yeah, and what cellular process gives off CO2? We kind of think of it as sort of the opposite or the counterpart to photosynthesis.
Respiration.
Yeah. So when we talked about the radish experiment, we saw that plants respire as long as they're active and not dormant seeds. They are respiring. They're using that sugar to maintain their cellular functions and survive. So what do we mean by a dark respiration rate? This is a curve for an individual plant, for example. It just tells you the respiration rate of the plant when it's not doing any photosynthesis. It just tells you what it's doing sort of at rest when it's simply consuming carbon dioxide and not photosynthesizing at all. And as soon as you add a little bit of light to the system, as soon as you expose that plant to some light, you think of it, this is dawn, right? The sun is just starting to come up, there's twilight. Eventually, some of those photons are going to be captured by the antenna complexes in the photosystems, right? And you start to get the Calvin cycle powered, you start to fix carbon, photosynthesis is happening, but if the rate at which sugar is produced is not enough to sustain the plant, you're still going to be in that negative space. So we've got a point here at which there's enough photons coming that respiration and photosynthesis balance each other out perfectly. That's called the light compensation point. It's the light intensity when you get sort of an equilibrium of carbon dioxide exchange, respiration and photosynthesis are balancing each other out. And then as you continue to turn up the light, as it gets brighter and brighter, the assimilation rate goes up as more and more carbon is fixed because there's more ATP and NADPH available to power that Calvin cycle. Eventually, the curve starts to flatten out. You get to an area where the system is light saturated. A little bit of an increase or even a big increase in brightness has a correspondingly small effect on the rate of CO2 assimilation. So over here, more light, more CO2 assimilation. The reaction is light limited. Over here, the reaction is light saturated. So if you add more light, it doesn't speed up the reaction. If something's limiting the reaction, it has nothing to do with light. It has to do with other parts of the reaction, maybe the amount of carbon dioxide available or something like that. So that's kind of what I want you to think about this question when you're going back over this. What do we mean by light saturation? It means that the system is no longer light limited. If you add more light, you don't get much increase in CO2 assimilation rate. So that's starts to get you thinking about what might limit the process of photosynthesis. If it's a great thing to produce all the sugar and plants can grow, there are certain conditions where you might add more to the system, but not get much return on it. So one of those examples would be at a certain point, adding more light doesn't make this go faster. So what else could limit photosynthesis based on this diagram besides light? Are there any
The
other?
amount of water available.
So that's a good answer. And it does limit plants' ability to grow. Like water availability does limit plants' ability to grow. But the amount of water that's necessary for light reactions happens to be very small. And it's transpiration that plants need water for. And we're going to talk about that in just a moment. So that's a good first guess. That's usually the first thing people think, amount of water. But really, there's not much water that's needed. And if a plant didn't have enough water to do this, it probably wouldn't be alive at all. What's another thing that might be an input to the system that could limit the rate?
ADP and NADP.
Yeah, so that's a good one, right? So the amount of these energy storing molecules that can be cycled between here and the rate at which they can be cycled is going to limit the system. And one of the important elements in this system is phosphorus, the core of those phosphate groups. And phosphorus is not a very common element compared to some of the others like carbon and oxygen and hydrogen and even nitrogen. It tends to be a limiting nutrient, and so there's only so much available to cycle back and forth between the two reactions. So you're right generally, but I would say it's the phosphorus really that can limit the rate of the system, the availability of phosphorus to make these molecules. There's one other, somebody else. One other input.
Carbon dioxide.
Yeah. So carbon dioxide availability can limit the rate of photosynthesis. Now you might be hearing that carbon dioxide concentrations are increasing in the atmosphere right now. We passed 400 parts per million a couple of years ago. Generally right now, carbon dioxide concentrations are relatively low for the history of the Earth. So it's been much higher now. It's been much higher in the past than it is now. And that actually is related to this C4, C3 concept that we're going to talk about. So let's look at CO2. How do plants get CO2? Well, you can think of plants as covered in cuticle, which is a waxy coating that is nearly impermeable to gases and to water. So to exchange gases, to release oxygen, to take in carbon dioxide, and to release water, plants have pores called stomata. And you can see the pores are surrounded by two cells, guard cells. So they have this characteristic look of like two little bean-shaped things surrounding an opening. That's a stomatal pore. As carbon dioxide is taken up through the carbon fixation reactions, the concentration of carbon dioxide inside the leaf goes down because it's being converted into sugar. So if the carbon dioxide concentrations are low in the leaf compared to outside the leaf, CO2 diffuses inwards. But as the stomata are open to allow that CO2 to diffuse inward, water is lost to the outside. So the uptake of CO2 from the atmosphere into the leaf is driven by a concentration gradient in CO2. There's also a concentration gradient for any air molecules that you might be interested in. For example, the water. Water is lost because it's humid inside the leaf and it's relatively dry outside of the leaf, so water is lost through transpiration. It's also the case that that oxygen has a concentration gradient. It's being produced in the leaf. And so when the stomata are open, it exits. Notice that the concentration of carbon dioxide is very, very low compared to the concentration of oxygen. There's a lot more oxygen than there is carbon dioxide on a molecule for molecule basis. So let's think about CO2 for a moment. Inside the leaf, intracellular CO2, carbon sub I in this case means intracellular, can be measured in concentration. And we can look at how this concentration affects the rate of carbon dioxide assimilation or photosynthetic rate. As the concentration of carbon dioxide in the leaf goes up, the rate of CO2 assimilation goes up. Eventually, you get to a point where adding higher CO2 concentrations into the leaf mesophyll doesn't really affect the carbon dioxide assimilation rate anymore. So it levels off. And this is where the photosynthetic rate is limited by the rate of the reactions inside the chloroplasts. So how fast can Rubisco fix carbon dioxide and make those 3-PGA molecules? And how much phosphorus is available to power the Calvin cycle? If the system is operating as fast as it can, given the rates of these reactions, then adding more carbon dioxide isn't going to increase the rate of the reaction anymore. So you switch from CO2 limited to a CO2 saturated condition. And we're thinking specifically about that little microenvironment inside the leaf where carbon dioxide concentrations might fluctuate. Now, the concentration inside the leaf is going to be related to two things. It's going to be related to the assimilation rate, the drawdown of carbon dioxide from photosynthesis, but also atmospheric carbon dioxide, the amount of carbon dioxide in the air around the leaf. That's what sets up that gradient that pulls carbon dioxide into the leaf. So what that means is that down here, a small increase in carbon dioxide concentrations in the atmosphere should lead to an increase in assimilation rate, all other things being equal. Now, in order to get carbon dioxide into the leaf, the plants have to have their stomata open, right? They have to have those pores that allow that gas exchange. When the stomata are open, they also lose water molecules through the process of transpiration. So why don't plants just leave their stomata open all the time and trade those water molecules for carbon dioxide molecules? Anyone have a guess? If keeping your stomata open means you keep that concentration of carbon dioxide in the leaf high and you keep the reaction of photosynthesis going, why not just leave your stomata open all the time?
Because then they will lose a lot of water. They cannot intake that much water from the soil.
Yep, good. So when the plants can't transpire enough water to match the evaporative demand of the atmosphere, the loss of water through the stomata, they wilt and ultimately they die. And because plants have to maintain a certain negative pressure inside themselves, and if the pressure gets too low because of really high evaporative demand, because of a lot of water loss, air bubbles start to form, cavitation happens, and the water column that keeps the plant alive breaks down, and the plant can wilt, lose leaves, or even die. So they have to conserve water. They have to balance their water loss with the amount of water they can take up from their roots. That's the key. They have to balance water loss from inside the leaf with their ability to replace that water from their roots. And that depends on things like how much water is in the soil and also how dry the air is around the leaf, how quickly is water lost to the atmosphere. So there's kind of a cost-benefit relationship then when it comes to opening and closing stomata. The more you have it open, the more CO2 you get, but also the cost is the more water you lose. And you can see in a typical C3 plant, like we've been talking about so far, for every 500 molecules of water loss, you might gain 1 carbon dioxide molecule. So you really are losing a lot of water. It's a high cost to what you get. So we describe this trade-off with water use efficiency. It's the ratio between the gain of carbon dioxide and the loss of water. That's what we call water use efficiency. It depends on diffusion, and that's a concept you should remember from chemistry. Specifically, it depends on the diffusion of water out of the plant and CO2 into the plant. Plants can increase their water use efficiency by decreasing the amount of water that they lose to the atmosphere through transpiration. And there's 2 ways you can do that. You can conserve water by closing the stomata, so you don't lose water to the atmosphere, or you could have increased water use efficiency if there was a higher concentration of carbon dioxide in the leaf. Plants can't really control the amount of carbon dioxide in the atmosphere, but they can open and close their stomata. So that's the main way that they control their water use efficiency. The challenge that comes with closing the stomata has to do with this enzyme Rubisco that I've talked about a little bit. Rubisco ends in the 2 letters C and O. The C stands for carboxylase. It's an enzyme that fixes carbon dioxide. And the O stands for oxygenase. The enzyme also has the possibility or the capability of fixing oxygen to this RUBP. So it prefers carbon dioxide, but about 30% of the time, it'll go for an oxygen molecule instead of a carbon dioxide molecule. And if stomata are closed and Rubisco is fixing the carbon dioxide, the concentration of carbon dioxide in the leaf goes down, 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 and the odds that the Rubisco will take an oxygen molecule instead go up, up, up. And the problem is when you have a 5-carbon molecule, RUBP, and you fix an oxygen, you don't get 2 PGA molecules. You get a 2-carbon molecule and a 3-carbon molecule, and that's not good enough to power the Calvin cycle. So this process is called photorespiration, and it's where you take in carbon dioxide with Rubisco, bind it to RUBP, and you get these 2 molecules, a 2-carbon and a 3-carbon molecule. This is what happens when CO2 concentrations are low because the stomata are closed, and it's a problem for the plants because they can't produce sugar. Notice that over here, sugar is produced, and over here, carbon dioxide is released. Right. So I think this might be our last slide for today. When is the risk of photorespiration highest for plants, for C3 plants? Well, this graph relates the concentration of carbon dioxide in the chloroplasts to the temperature in the environment and the ratio of photorespiration to photosynthesis. Are you more in that yellow cycle or are you more in the green cycle? If you're more in the green cycle, more in the photosynthesis cycle, you're down here in the blue, right? A low ratio. And if you have a lot of photorespiration going, you're going to be up here. So under what conditions of temperature, what combination of temperature and CO2 concentrations results in a high rate of photorespiration? Can you guys read this 3D graph and tell me that?
Is it high CO2, low temperature?
That's where you're down. So high CO2 and low temperature would be down in here. This is going to be blue. So this is where you've got this is where photosynthesis happens and it's okay. You're getting that Calvin cycle powered. If you go out here to high temperatures and down here to low CO2, you're in this back corner, right? And in the back corner, that's where you're up here in the red with high risk of photorespiration. So I'm not sure you got that incorrect. I might have asked it a little bit weird. because this is a ratio. But what we want you to know is that low CO2 concentrations and high temperatures in this back corner, that's where the risk of photorespiration is highest. Hot soils, low humidity. So I'm running a little bit behind, but we're going to talk about next time is the solution to this problem, because conditions where you have low internal CO2 and high temperature are actually pretty common. Common in the summertime in the prairie, common throughout much of the tropics. This is a problem that plants have to deal with. The main problem is low CO2, and the solution is a mechanism for concentrating carbon in the leaf, and we call that C4 photosynthesis. So we will start with this slide on Friday, because you have advising day on Wednesday, and we'll talk about the difference between leaves that use C4 photosynthesis and concentrate carbon dioxide versus leaves that do C3 photosynthesis using the typical system that we just talked about last time and today. Any questions as we come to the end for today?
Okay, look for an email from me about times when I want to meet with the various groups tomorrow in lab, but we will be having lab tomorrow, and that's when we'll go through your workshop slide presentations that you've prepared, but we'll do that with each individual group, so you'll be given a time and look for an email from me.
Great. All right, we will see you guys, well, I'll see most of you on Wednesday, but I'll see the rest of you on Thursday.